So this is our second video on DNA, our last video before we really get into protein synthesis. And this is on DNA replication, so how DNA copies itself. So the first thing we need to know the when and where. So when DNA is only going to replicate itself during the S phase of interphase. So if you look over here, this should be your cell cycle, which should be very familiar. We know that interphase right here is made up of G1, S, and G2. G1 and G2 are really growth and checking the cell to make sure that it's ready before it divides. But S phase right here, this is where DNA replication occurs. Where that's going to happen is going to depend on what kind of cell we're looking at. So if it's a eukaryotic cell, like this one over here, DNA replication is going to occur inside the nucleus. Now your prokaryotic cell, which you remember are your bacteria, they don't have a nucleus. So because of that, they are going to replicate their DNA just in the cytoplasm. Now, why would your DNA need to copy itself? So DNA is the instructions to make all of the necessary proteins for a cell to function. The proteins are really the bread and butter for your cells. So they're the structures of your cells, how they make themselves and how they run themselves. Daughter cells are going to need a full set of instructions so that they can run themselves and be fully functioning cells. So in order to form fully functioning daughter cells, we need a way to copy this DNA, this code, to carry on the function of a cell. So we need to know one more thing about DNA structure. So we know that DNA is a double helix. It's got two sides to it. So we know that the outsides of our DNA are our sugar phosphate backbone. So here's our sugars right here, and there's our phosphate groups. Now, the two strands are anti-parallel, meaning that they move in opposite directions. So you'll notice if we just take a look at this side over here, we notice up top we have what's known as a 5'. This little symbol right here means prime end. And all the way down here we have a 3'. Now this notation is basically showing you, it's a notation for carbons. So right here they're saying that your 3' is referring to the fact that you have an OH group coming off of your third carbon. So here's 1 carbon, 2 carbons, 3 carbons, so your 3' carbon. Up here, your phosphate group is coming off your 1, 2, 3, 4, 5 carbons. So there's your 5'. And you'll notice this is the opposite. So where on this strand we have our phosphate group pointing towards the top and our OH at the bottom, it is the exact reverse on this side. And that's what anti-parallel means. We have our OH group down here, which is on the top, and we have our phosphate group on the bottom. So they move in opposite directions, although they are complementary. Now, DNA, when it's replicated, is only added to the 3' end. So when these strands divide and new base pairs are added, on this one, it's going to be added down here at this end, but on this one, it's going to be added up here on this end. So how DNA is replicated? Well, after the discovery of the structure of DNA, there did not really answer all of the questions we had about DNA. So we knew that it was the code for how proteins were made, but we didn't know how it copied itself. So there were 3 basic models that people had. The first one was the semi-conservative model, which told us that we have 2 original strands that divide, and our new cells are going to use one as a template and one as a new strand. But then there was also people that believed in the conservative model, which meant that your 2 strands serve as a template, but they're going to stay together and you're going to get 2 brand new sets of DNA. And then finally, the dispersive model just said that these are going to chop up. So you're going to get little pieces of new in this one and little pieces of new and old in this one. So it said that there's no distinct pattern. Now, there was a neat little experiment done with tagging the DNA with isotopes of nitrogen. So they were able to actually follow the percentage of these isotopes as they move through DNA replication. And what they found is that the semi-conservative model is what we actually have. So what semi-conservative means, as I said, is that your DNA strands are going to split. So we know we have a double helix and 2 sides. So they're actually going to split down the middle, and each half is going to serve as a template to form a completely new strand of DNA. So that's how we're able to conserve the code, so keep the same code going through multiple replications. So your replicated strands are going to be half original DNA strand and half new DNA strand. So a little quick check-in for you guys. So we're going to go through DNA replication, but here is my strand that I'm starting with, my template strand. So I have adenine, cytosine, guanine, adenine, thymine. See if you can tell me what the complement is to this strand of DNA. Good, so hopefully you were able to get that is this sequence here. So remember, adenine and thymine to always pair up, and cytosine and guanine always pair up. So here we have our whole parent strand. They're going to split during replication. Your nucleotides, so the new ones are going to be coded in orange here, are going to come in. They're going to fill in and bond with their complements, so they keep the same pairings. And here are your two identicals that are half old, half new. So the MVPs, most valuable players that you guys need to know, are enzymes. And the two enzymes that you're responsible for knowing, the first one is helicase. So remember, this is the double helix. So it has two sides to it. So helicase, ASE tells us it's an enzyme, is an enzyme that unzips the double helix. So what it's going to do is come in and it's going to split those hydrogen bonds that keep your nucleotides held together. So it's going to break up those base pairs so that those new nucleotides can come fill in and really form that, replicated strand. The next one you need to know is DNA polymerase. Now, there is an RNA polymerase. When we talk about RNA, we'll get into that one as well. But polymerase is really the most important enzyme. It's the enzyme that adds complementary base pairs. So it's actually going to find what base pair belongs with the next one, and then it's going to add it to it. And it also proofreads. So if you look up top here, so this is our DNA polymerase. And so we have an adenine right here. Thymine's going to move in. DNA polymerase is going to add it. So it's actually going to do the little chemical reaction to make that stick there. And then it is going to proofread. Is that right? Yep, we're all good. So here, if we have an error, your DNA polymerase is going to recognize the error. It's going to kick that wrong nucleotide base pair out, wait until the right one comes along, and then add that one and keep going. And see how it's moving? It's only adding them at the 3' end. So it's adding them at 3' only. So that was pretty much it for DNA replication. Just a little quick and easy overview. So comprehension check. First one, where does DNA replication occur in prokaryotes? So hopefully we didn't go with nucleus on that one, because we should know by now that our prokaryotes or bacteria do not have nuclei. So that means that DNA replication for them is just going to occur in the cytoplasm. Number 2, DNA replication is good. It's semi-conservative, meaning that each of your strands are going to split using helicase. It's going to drive up the middle of that double helix. And each half is going to act as a template and be filled in to form 2 new identical strands. Number 3, what is the function of DNA polymerase? Good. Hopefully you got that it is a proofreader, and it also adds your nucleotide base pairs. So hopefully you were able to get those. All right, make sure your notes are filled out and message me if you have any questions.